Okay, hello. This is round 2 of the lab discussion videos. Cameron Bocanegra here, if you recognize my voice, or maybe just the fact that my name is on the screen. I chose the Carbohydrate Fermentation lab for this video. It's most interesting, and I just felt like doing it, so let's get right into it. Introduction. So basically, in this lab, we tested 3 different substances: fructose, glucose, and mannitol for fermentation. The main question was, if I had to put it in words, how are different sugars metabolized by yeast? It was in a controlled environment. We had the sugars and we had the vials of phenol red broth, and pretty much all you do is mix in some yeast with some water, pour it in there, put the sugars in, and you put the Durham test tubes in there. The test tubes test for gas production because the bubbles will kind of build up inside of them, and the phenol red tests for acid production because it'll change color based on how much is being produced, pretty much. And then we also had a control vial which only had yeast in it, just to compare amongst the different sugars and show what would happen if you didn't have anything accelerating that fermentation in there. My hypothesis, I said that that glucose would see the highest amount of fermentation because it's a more, not necessarily fuller, but I'd say it's a more complete energy source when compared to fructose and mannitol. Fructose is a lot simpler, and mannitol is a sugar alcohol, so they may be metabolized a lot quicker or slower, just depending on which sugar you use. Since glucose comes from more condensed sources such as starch, I predicted that it would be able to produce the most fuel and would ferment the most effectively out of the 3. The data. The data was collected in the form of a table and some pictures. This photo right here is just to show you what the phenol red's supposed to look like before you add anything to it, since I didn't get a picture of it before I added the sugars or the yeast. It's called phenol red for a reason. The photos were taken after 12 hours of testing. This is just to show you comparison. Let's move on. Here are the results. This is my lovely table. As you can see, just denotes which vial you have, the color change, gas production, the conclusion. So as you can see, fructose, all of them changed color except for the control. Glucose and fructose turned yellow and mannitol turned orange, and glucose had the most gas production. Just because it says N doesn't necessarily mean there wasn't any. I just picked it because there wasn't nearly as much as in glucose. You can read over that if you'd like. Then here's the photo. Apologies if it is a little blurry. Didn't get the best angle. But as you can see, here's the glucose, fructose, I don't know why I put an E, mannitol, and here's the control group. As it said in the table here, glucose and fructose turned yellow, mannitol turned a more orangey color, and the control maybe turned a little lighter red, a little pinkish, but you can see a little bit of orange on the bottom there, but it pretty much stayed the same. And this was after 12 hours. I'll reiterate. Oops. Okay. As shown in the data, glucose was the clear winner. You can't really tell in this photo, but there were a lot of bubbles in the Durham tube in there. Not so much in these other 3. A little bit in the fructose, a little bit in the mannitol, not really much in the control. It also had the highest color change, pretty striking yellow. This one's a little cloudy. And then these 2 closer to pink and orange. Fructose fared almost as well. As you can see, it showed clear signs of fermentation with the yellow color change, but it didn't have as much gas production as glucose. Mannitol only changed to orange and only had a few little bubbles in there. And the control didn't really have anything in there, and it only really turned a little lighter. So, glucose wins. Hooray. Conclusion and discussion. As you can tell, my main conclusion is that glucose was in fact the best facilitator out of the 3 options that we were given. Showed the most signs, showed the best color change, had the most bubbles in it. Pretty simple. This aligns with my hypothesis because that's pretty much what I said. I predicted that glucose would be the best out of the 3. So, practical applications. I could see this being useful for businesses where you're using fermentation like a brewery or a bakery. If you're running one of these businesses, you would want to know which one, if you would want to add glucose or fructose, if any, to facilitate fermentation so that you can make your product more efficiently and more effectively. And that's pretty much it. Thank you for watching, and enjoy yourself.